Welcome to Whose Genome Is It Anyway? Big Data and Your DNA. I'm Sarah Norcross and I'm the Director of the Progress Educational Trust, PET, and we're a charity that aims to improve choices for people who are affected by genetic conditions and by infertility. And we do that by encouraging, in whatever way we can, the responsible application of science so that it can benefit both the patients and the public. And we do this in a number of ways. We do it by leading the debate, so by identifying the challenging issues in these areas, which is quite an easy job because there often are lots of them, and by putting on events like this to encourage a discussion about them. Influencing policy. This is really important to us, so that it's better aligned with science and that it can reflect what society wants the science to do, and that the scientists and the public can both benefit from that. And then by informing and explaining. So one of the things people know us best for is BioNews, our weekly email news digest. And also by putting on events like this and doing other outreach activities. So thank you very much for coming along tonight. We're really pleased to see you. And we're delighted to be working with Genomics England as part of the Genomics Conversation here in Scotland. And it's really important with projects like this to get lots of people involved and talking about them and understanding what's going on, because there's plenty going on and there's lots to learn. So we're delighted to be in here in Edinburgh, and I'm delighted to introduce tonight our chair, Professor John Mattick, who is the Chief Executive of Genomics England. And he has an international reputation as a scientist. He's published nearly 300 scientific papers and has won many awards. He's got more citations than there are Genomes Projects or Genome Sequence, probably. And I was looking online to see what John had said about the sort of issues that we're discussing tonight. And the internet is a wonderful thing because you can find all sorts of quotes for people. So this is what John said. He said, "I think that people want more control over their personal data and more access to it. It drives me crazy that I don't have all my personal records on my smartphone and I can't walk into a physician and have them download it." We're talking about a new data ecology that will be person-centric and one in which current data structures aren't suited to serve. So he clearly has views about how he would like his data to be used, which may or may not tie in with the project because they're his personal views. Anyway, I can see John thinking, oh no, why did you say that? So I will stop there and encourage you to give John a very warm welcome this evening.
Thank you, Sarah. They are my personal views, but I think they'll come to pass and be part of the discussion this evening, I think, because to answer the question, whose genome is it anyway, is your genome, of course, and my genome. And the question is, how do we access it and who do we allow to access it, for what purpose? I'd like to add a question before I start to rub out England off this slide. Add another question, if I may, Sarah, and that is, would you be happy to have your personal genomic information used not simply for research, but also for your clinical care, your health care in the future? And let me explain that, because I think that's where we're going. And I won't speak for long, it's just introductory remarks, but I've been in the business for a while, and I can say with some confidence that there's hardly any aspect of human biology which is not profoundly influenced by the idiosyncrasies of our genetic heritage from our ancestors. And if that is true, then understanding the details of those idiosyncrasies will open up a much more profound understanding of human diversity, human talents, human perspectives, human disease, etc., which will, I think, be a golden age for discovery. Finally, I think we'll start to understand who we really are as individuals and as a species. Up until now, you may be aware that most of medical research has been both reductionist and very heavily dependent on model organisms, and they've been very useful. I mean, yeast has taught us about cell cycle. Many of the genes that yeast has to control cell division are actually analogues of ones that we have and are involved in cancer. Funny model organisms like fruit flies and nematode worms in mice. But now we're going to be able to study humans. We're going to have millions of genome sequences, high-resolution genetic information, most of which we don't yet understand, and millions of matched clinical records and information that will flood in the coming years from smart devices that you may wear. And of course, so this whole ecology of both genotype and what we refer to as phenotype will come together, both to inform our understanding of humanity and human diversity, as I said, but also to inform clinical treatment. And I think that where I would like to take this, and in partnership with people in Scotland, is to progressively bring it into the clinic, and this is going to be a big challenge for all of the structures around the health service in this and other countries, because the model for healthcare at the moment is based on crisis management and delivery of care at the point of crisis. And we are going to move through the information that floods in from our personal, looking under the hood at our personal genomes, as well as other information about us, to a changing paradigm where the health system will be geared more strongly and progressively towards health management and maintenance. For those of you who haven't read the book called The Patient Will See You Now by Eric Topol, and the NHS in London is doing a review, Topol review, I recommend it. It's not expensive. Every chapter is an epiphany. For example, patient empowerment. Many patients know more about their conditions than their treating physicians because they've got the incentive and the time to investigate. The changing nature of hospitals from crisis centres to wellness centres. The impact of smart devices and that information, as well as, of course, of genomes. So I'm looking forward to a future where genomic information will reveal which ones of us in this room are at high risk for cardiovascular failure. And there are ways, of course, as intercede earlier, to mitigate that risk or prevent it. Which of you are at high risk for cancer? That's a recent study we carried out with the National Cancer Institute in the United States so that we could predict who had cancer more accurately than any other technique just from genome sequence. And gave those individuals MRIs, and 7% of them actually had pre-symptomatic cancers, which were easy to treat at that point. So I could go on, but I think it's the fundamental foundation of this coming revolution in healthcare. And finally, I think we have in sight the holy grail of healthcare, which is to use high-resolution information about us, both genetic information, genomic information, and physiological information, to actually introduce personalised and precise medicine and healthcare, which will be much more effective at keeping us healthy and creating a healthy society and a much more sustainable and high-quality healthcare system. I'd like to introduce the first speaker. I should say we won't be taking questions after each talk. We're going to save the questions for the end and make them more discursive. So there'll be a series of 10-minute presentations. I have to stand to attention if people transgress the time limit. And hopefully they'll be entertaining, I'm sure they will be, and then we'll go to questions and discussion. Now, I have to look at my phone because this first speaker, I just got her first name right, Zosia, but then I realised that she's got a very long, multisyllabic, I presume Polish name, Zosia. So forgive me, ladies and gentlemen, Miedzybrodzka. Is that close enough, Zosia? I hope you'll forgive me. Zosia is a professor of medical genetics and the co-director of the Centre for Genome-Enabled Biology and Medicine at the University of Aberdeen. Zosia.
So thank you for having me here today. I walked in today and this morning I did my clinic, which I do a couple of times a week, and I see patients. I'm a doctor who has trained in genetic science, who is director of a laboratory, but I see patients too. And I saw a patient this morning who had a 1 in 4 risk of an inherited condition. Now, as we talk today, we've been hearing about the high-level stuff, and I'm going to walk you through what we're doing in Scotland, working with Genomics England. But I think it's helpful to just set a context for where we are in Scotland today. I saw an educated person at 1 in 4 risk of an inherited condition, and they did not know how DNA worked. So I was able to introduce that because of building blocks they had, but they still really didn't understand even the very basics of DNA. When I wrote my talk, I wasn't sure what basic underpinning people in the audience would have. So I've got a few slides explaining the kind of things I explain to my patient. I'm here speaking on behalf of the Scottish Genomes Project, which encompasses a number of different aspects of genomics, and the project started as a collaboration led by Tim Aitman, who's here today, and from the University of Edinburgh, Andrew Biankin from the University of Glasgow, and it followed really from a major investment by those universities in equipment so that we could, for the first time, sequence a genome, that's all of somebody's DNA, instead of just little bits of it. And that project was funded by the Chief Scientist Office, by the Medical Research Council, in order to build on Scotland's strengths in genetic research, but also in clinical practice, and to take the genome test from a research test into the clinic and work out when and for whom we should be using it. The Scottish Genomes Project has actually many aspects. There's a whole, in rare disease, there's a whole piece on population cohorts. We're sequencing, colleagues are sequencing people from the Viking cohort in Shetland. And there's also a lot of work on trying to discover new genes for rare disease and motor neuron disease. There's a major program of work on cancer. And I'm not going to talk about that because today's talk is focusing on using people's DNA and using their data about their own care. And I think it sits best with the bit of the project I know most about, because it's the bit I'm leading, which is about patients in NHS Scotland taking part in the 100,000 Genomes Project, which started with Genomics England. So we're focusing in Scotland on rare disease, and the project is run across 4 centres. Genetics in Scotland started working together in an organised way 32 years ago, and that's how we deliver genetic services, both talking to patients in the clinic and through labs. And we're very generously funded, by the National Services Division, who are here tonight, whose job it is to make sure that every patient in Scotland has access to the same tests, and to make sure it's done in as cost-effective a way as can be done for the benefit of patients in Scotland. And I focus on costs a lot. They say an Abbotonian, where I now live, is a Scotsman with a generosity squeezed out. Now, I've worked in the NHS for too long to know that you shouldn't try and do anything that you can do cheaper. So the NHS is very much about making the money you've got spread as far as you can over as many patients as you can. And the context in Scotland is that at the last time we looked, in 2015, Scotland did something like 25% more genetic tests than were done in England. And we don't even know how many Wales did because they didn't count, or at least they didn't tell us. So Scotland is a nation that does genetic tests in the clinic. But so what is different about the concept of a genome? Scottish Government's policy had a focus on identifying and preventing rare disease in rare disease policy. And that is, genomics is going to be the key to that. DNA is the chemical in our bodies that's the program to make proteins. We have 22,000 genes, and they are very different from each other. And it's, for those of you who are aficionados of these, will know that DNA is not just about the genes, which actually make the proteins, but there's lots of bits in between that we're only just starting to understand that control whether those genes are switched on or switched off and how switched on they are. And the real key about a genome test is it can tell you about all of that. The current paradigm in the health service is we only look at the coding DNA because we can start to understand that. But in the coming years, we're learning faster and faster about all the stuff in between, and that, I think, is where the real goal of this project is. But what are we actually doing in the clinic today? Most genetic testing remains the cheapest chips, standard test where we focus on the gene that the person's interested in, or maybe now we're doing 5, 10, even 100 genes at a time. But there are something like 10,000 genes in rare diseases. And there are groups of conditions where we don't know where to start looking. And at the moment, that's the place we're really focusing the genome at. And it is really like looking for a needle in a haystack. If you don't know where the needle is, then you have to be able to find a systematic way to look through it. And the key to this is data. So, for example, we have about 6.5 million variants in our DNA. This family had a child with a learning disability and they didn't know what was wrong with her. So they sequenced her DNA and the parents' DNA. So they found all those variants and which one was the problem. Well, what we were able to do, because by comparing her DNA with DNA from many, many people, not just in the UK, but all around the world, was work out which 670,000 odd variants were actually incredibly rare. And then filtered those throughout all the ones that didn't actually change anything in the way the genes were read into protein, and then take those and focus down on, well, what did she have as an alteration that her parents didn't have? And then that still left 67, but only one of those was a gene known to cause learning disability. That was the GLUT1 gene, and there's a treatment for it with a ketogenic diet, and that child improved with that diet. So this is the paradigm, and it's only the data, and it's only having the background data that lets you do that filtering process, tells us where we are. So if we want to change people's healthcare with genomic data, we need lots and lots and lots of genomes to look at, and we need to be able to compare them, and we need to know what's wrong with the people. So that's the reason why everyone being asked to take part in the Genomes Project has been asked to share their data, both not just now, but for the future. And through electronic records, that can be done in an automated way. And Scotland's contributing to that. But a key difference in Scotland from England, we've done 1,000 genomes. Patients have consented for the data to be shared. But at the moment, the cipher for which patient is which in the database is held in the genetics centre. So patients in Scotland's data is identifiable to the genetics centre, but not to anyone looking in at the key NHS database. But researchers are able to look at that database by taking away all the identifiers. And that is now becoming one of the largest genetic data sets in the world for research. So what we're doing in real life is comparing those data and taking them and then giving patients results that will change their care. Today, at 12 o'clock, we put the 1,000th patient into our study. So what we're going to do in the future is report back results to all of them, and hopefully those will be important results that will be useful. And as we evaluate that, we'll see what the place of this current system is in today's technology. Thank you.
Thank you, Zosia. Our next speaker is Dr. Austin Diamond, who is the Quality Assurance Manager at NHS Lothian's Genetics Service. Molecular Genetics Service. I'm sorry, Austin. Welcome.
Okay, so I work at the sort of sometimes what's called the sharp end of genetics. So where we actually get the results back from genetic tests and we have to interpret them. And this is a big problem because, as Zosia indicated, there's a huge variety of genetic disorders. This slide also makes that point. It also makes a second point, is that genetic disorders are quite common. They affect up to about 4 or 5% of the population, which also means, conversely, that 95 to 96% of the population don't have any genetic disorders. And that's also very useful information to us, and I'll show you why in a little while. We're obviously asked to do genetic tests. Now, if you're going to offer a genetic test to somebody, you've actually got to know what to test. Now, the only way you can actually do a useful and valid genetic test is to have some handle on the result. And that means if you want to say this person will get that disease or this person won't get that disease, you've got to know which variants in DNA will cause disease and which ones won't. So all this testing is predicated on understanding what variants actually mean for the patient. When we find a variant, as a lab, we have got to put it into one of 5 boxes. All variants start out in this box in the middle, a variant of uncertain significance. And then what we have to do is we have to go and look for evidence that allows us to move the variants either up the scale to likely pathogenic or pathogenic. And if we can put them up there, that means we can use it for testing. That means we are confident to some extent that they are responsible for a patient's condition. Or we can push them down between likely benign and benign and say, actually, that's fine. These are just variants that are found in the general population and they're nothing to do with that parent in that patient's condition. So we have to do this in an evidence-based way. And a few years ago, in order to ensure some consistency, so if you have a test done in Edinburgh and your cousin has a test done in London and they both find the same variant, it'd be really nice if both labs actually reported the variant back in the same way, they put it in the same box. The ACGS, the British Society, decided they were going to adopt the American guidelines for how to do this. Now, the American guidelines look all terribly scary. There's this sort of massive, great matrix of things, and down the left there's a whole lot of different sorts of evidence, and going across from left to right there's stronger and stronger indications that they might be pathogenic. I regard these things as sort of giving variants, these bits of evidence, like what I call pathogenicity points. And when we get a variant, we have to decide how many of these boxes it goes in, and for each box a variant can be put in, we award it a certain number of pathogenicity points. And then we add all these pathogenicity points up by a set of nicely defined formulae that are given to us. And the idea is that when we do all this properly, every time anyone interprets the same variant, they're going to get the same answer. They're going to put it in the correct one of these 5 boxes. So are we all happy with that as a concept? So all your variants go to one of 5 boxes. These are the sorts of the types of evidence that the ACMG thought we would be able to use. In practice, there's only really 2 of them that come up very frequently, and those are population data and computational and predictive data. And all the other types of data really aren't usually available for most of the variants we're dealing with. I want to focus primarily on population data. These are the ACMG criteria, and I'm going to translate those for you. There's 2 criteria that say you can ignore this variant, it's not really relevant. The most common is this first one, saying that far more people in the general population have this variant than have the disorder. So this is a common variant, nothing to do with disease. There's a subtle variation of that, which is people that you know don't have the disease have this variant more significantly than it would be expected if causing it. Those are more or less saying the same thing, either for a general population or a selective population, that you find the variant more often than the disease. And then there's 2 things that suggest it might be pathogenic. The first is it's not present in the general population, and even more important, if it's significantly increased in an affected individual compared to people who aren't affected. So these are sort of the main lines of evidence we're trying to find. Now, luckily, a couple of years ago, a huge data set was released called the NOMAD data set. And the NOMAD data set has the sequences of all the genes of 140,000 people, to a quarter of a billion variants. These people are from around the world, and they're not a completely random population. They are selected to have a low frequency of early onset and obvious diseases. So if someone is born early and they showed symptoms in life that are consistent with genetic disease early on, they'll be excluded from the NOMAD data set. But the other thing to remember, and I'll come back to this, is that if you have a disease later in life, you may still be in this data set. So it's not a control data set, and it's not actually population data. It's sort of like a hybrid between the 2. The control data set being, of course, one where there's no disease, population data set being everybody. So I want to now actually look at some proper data. So this is an example of what we find when we tested an individual who had microcephaly. You probably all remember microcephaly from the Zika virus outbreak in Brazil a couple of years ago. Now, a lot of microcephaly is like that. It's caused by something environmental or sporadic, but a small proportion is genetic. So we've tested a child with microcephaly, and we found a variant in one of the genes that is known to be implicated in genetic microcephaly, a cell called KIF11. So how might we decide that this variant is pathogenic? Well, one piece of evidence would be, as I said, it's not present in the general population. And if I look in that top box, that shows the frequency of this variant in the nomad population. And as you can see, that's a totally blank box. This variant has never been seen in nomad. Now, there's two ways that could happen. First is it's generally not been seen in any of the 144,000 people who've been sequenced, or that bit of the gene wasn't sequenced. So we had a quick bit of check. And you have to trust me on this one, that this is the bit of the gene that the variant would have been in had it been present. That's been this is a coverage plot, for those in the audience who understand these things. Lots of coverage. KIF11 is a very variable gene. There's 1,300 variants in KIF11 in the general population, but this variant isn't one of them. So this is a very rare variant. It's also known to affect the gene function, actually very damaging. Again, for those in the know, this takes out a consensus splice donor site. For everybody else, this basically wrecks the gene function totally. So we have something that wrecks the gene function and is very rare in the general population. So this is a really good candidate for causing that child's phenotype. A slightly more difficult case now. This is a variant we found in an individual who's got hypertrophic cardiomyopathy. Now, hypertrophic cardiomyopathy is relatively common. About 1 in 500 people have hypertrophic cardiomyopathy. Again, it's not always genetic, but it is sometimes, and there are half a dozen genes that commonly cause it, and another 10 or 20 that cause it far more frequently. In this case, we found a variant in one of the genes that commonly causes hypertrophic cardiomyopathy, MYBPC3. Now we look and we see, actually, in this case, when I look and see, there's clearly people in the general population, in nomad, who have this variant. So does that mean it's not a pathogenic variant? Here's the total data from nomad again. And if I look in Europeans, actually, this variant is really rare. There are 3 people in Europe out of the 55,000 people who had their gene sequenced who have this variant. So it's really not common in Europeans. But if you look up a little bit, it's actually quite surprisingly common in East Asians. In fact, about 1 in 500 East Asians carry this variant. Now, there's 2 possibilities here. Either there's a lot more hypertrophic cardiomyopathy in East Asia, and this is a common cause of the variant in East Asians. Or, actually, it's a common variant in that population and doesn't cause hypertrophic cardiomyopathy. It would be really nice if we had access to any health information about these 20 people, but we don't. It's a totally anonymized data set. But luckily, we have a little bit other information that, at least in a Southeast Asian population, hypertrophic cardiomyopathy is actually much rarer than in a European or Western population. So this is actually good evidence that that variant is not actually pathogenic at all. So I've gone through 2 examples where I've used the data from the whole population to help me classify 2 variants. I'm going to skip this slide. This actually shows exactly the same sort of example that Zosia has shown before, how you can use this to filter right down. And I'm going to come on for the last time to look at, say, what happens, what about variants in the other 5% of people that have genetic disease? Why are they particularly important? This is another gene that causes hypertrophic cardiomyopathy, MYH7. And an expert group have decided that you can adduce a certain amount of evidence from how many people in the world have the disease and have the variant. So it's strong evidence if you can find 15 people with the variant and the disease, and supporting evidence if you can find 2, and moderate if you can find 6. Here's a variant we've identified in a patient. If I look in nomad, it's actually a very rare variant. If I go to another of my favourite databases, this is the ClinVar database. Now, ClinVar is a place where labs can deposit genetic information along with the information about the disease that person has. So it's a variant and disease-linked database. We find that actually this variant is present 12 times in the clinical database in patients with hypertrophic cardiomyopathy. So here, this is now very good evidence that this variant, which is very rare in the general population, is enriched in people with hypertrophic cardiomyopathy and is therefore likely to be a pathogenic variant. So I hope the point I have made here is that having access to both general population data and also genetic data linked to disease incidence is incredibly useful for us. At the moment, we're working at as Zosia said, we're working at very small scale with just tens or hundreds of genes, but even so, we're still using this sort of inference. As we're scaling up into whole genomes, this problem is going to be really magnified, and this is why I'm really excited about the prospect of these very large data sets like the 100,000 genomes becoming available to us, so we can have huge amounts of genetic information, but crucially linked to whether the patients do or do not have the diseases which those variants may be associated with. That's all. Thank you very much.
Thank you, Austin. I might just follow Austin's remarks by saying that identification of the pathogenic variant for children, usually children, but also many adults, is extremely important because it gives the answers that families are looking for. It gives reproductive confidence because you can avoid that problem if you know what the problem is in the family, and in some cases you get transformative outcomes. Children who are literally in intensive care now at school. So it's the identification of the rare allele is the first step towards understanding and treatment. I might also just say quickly that we've sequenced several thousand individuals who've got to the old age, 80 or more, completely healthy and cognitively sharp as a tack. And there are rare variants in that population that seem to associate with both longevity and good health. So there's an upside as well to these variations. Our next speaker is Natalie Frankish, who is the Policy and Engagement Officer for Scotland for the Genetic Alliance UK and Rare Disease UK. So maybe Natalie will expand on some of these themes. Thank you, Natalie.
Thank you and good evening. Thank you very much for allowing me to come along and present a little on the patient perspective on tonight's topic. I'll start with a few words about Genetic Alliance UK, some of the work that we have done on data sharing and genomic sequencing. So for those of you who don't know, we're the national charity working to improve the lives of patients and families affected by all types of genetic conditions. We're an alliance of over 230 now patient organisations, and our aim is to ensure that there are high quality services, information and support available to all who need them. I should say that Genetic Alliance UK is also home to the Rare Disease UK campaign. And this is another rare disease community where we bring together the voices of patients and families and really all who support them to produce some high quality policy documents for bettering services and bettering policies for rare diseases. We also run the support network SWAN UK, which is a support network dedicated to families with undiagnosed conditions. So tonight I'm going to talk a little bit about some of the projects that we've done looking at genome sequencing and what patients think. So there's two patient charters, one which we published in 2015 and another we published in 2016. What I would say is I can't go into a great deal of detail in my 10-minute slot today, but you can read both of these online on our website. And I would also say that although they are two specific reports, actually the findings across both are very similar. So some of the key findings I've brought together here for you. These include the perhaps unsurprising view that patients want the option to receive as much information about their health as possible from genome sequencing. When it comes to the consenting of the use of their data, patients prefer a model of dynamic consent, something which they can opt in and opt out with based on how they feel, and they can readjust their preferences as things go on. This indicates that patients and public may feel more comfortable about allowing their data to be used for research if they knew that they had the control and the ability to change their preferences as they saw fit. Equally important to patients is the purposes of sharing their data. In particular, the requirement for it to have some benefit to the furtherance or understanding of a condition, developing new treatments or care pathways, or indeed shortening the diagnostic odyssey, that time to get a defined diagnosis. Another thing of value is information and support throughout the process of genome sequencing. and information about how data will be used. The two key findings that I'm going to look at tonight are firstly that patients welcome the sharing of their genomic data for research purposes. And secondly, it really is some issues around trust and about who can use that data. So as these quotes illustrate, patients typically display a very positive attitude to sharing genetic information for the purposes of better understanding of their condition. But not just their condition, also for others who maybe come along a little later in the journey. So it's often not just a case of a patient wanting to know about their condition, it's about making life better for who comes along next. So I'll let you have a little read of those while I talk on. But I should say across the surveys that we had, 93% of respondents to our surveys were happy for their genome sequences to be used for research purposes. And this is perhaps unsurprising given the patients that took part in this subject. For patients in the rare disease community, the need for patient data to be shared and used widely is not a new concept. Given the small numbers of individuals who are affected by a rare condition, patients recognise that collecting as much data as possible is really important. And often this needs to be done across international boundaries. It's vital if enough information about their condition is to be made available to research in order for them to carry out any meaningful studies. For these patients, not sharing data would be detrimental to research efforts and as a result to the scientific achievements these patients and families look to for the improvements to the quality and length of their own lives and the lives of their loved ones too. However, it's trust that's key about ensuring patients continue to feel enthusiastic about sharing their information and continuing to be willing partners in research. So moving on to this slide, our research has shown that patients and families' views on sharing their genomic information for research was very much dependent on the type of organisation that would be undertaking that research. In our 2015 report, over 2/3 of respondents to our survey were really supportive of the NHS, the universities and of charities for using their data for research purposes. And these figures stayed pretty stagnant in our 2016 report too. So around 80% for the NHS, 77% for universities and 63% for charities. And the fact that the majority of respondents supported genomic research within the NHS is a clear endorsement from our patient community that they welcome the current and the future initiatives that put the NHS at the heart of this type of research. However, it is worth considering some of the ways in which patients' confidence could be shaken. So media stories that show data breaches or perhaps the selling of third-party information without consent, these are things that can shake this confidence. So to maintain patient confidence, the NHS and the other bodies must take steps to enable the safe storage and sharing of patients' genomic data. And they also must keep patients informed about how they are doing that, providing information in a transparent way. So when it comes to trust, our research does show one contrast in confidence when it comes to private companies. In our 2015 report, only 38% of respondents said they were comfortable with private companies using their data. But it should be stated that in that report, there wasn't a definition of what private company was. The main reason participants gave for their disinclination to share their genomic data with private companies was that they weren't comfortable with the fact that some companies had the potential to make money out of that data that they were sharing. And this is quite interesting, particularly when we consider some of the recent developments and the rise that there has been in private companies doing genetic testing, like direct-to-consumer genetic testing. In our 2016 report, however, when we teased out the definition of private companies, we saw a fluctuation in response, with 61% saying that they do trust pharmaceutical companies with the use of their data when it's for a clearly defined purpose. We also gave our respondents an opportunity to answer whether or not they would be willing to share their genetic data with a private company if there was no explanation on how that would be used. And out of our 63 respondents, only 3 were happy for that to happen. And this may suggest that patients are comfortable with private companies accessing their genetic data, but only if that use is clearly defined. And the goals and aims of the studies run by such companies are made clear. Another point of interest, I think, from quite a lot of the patient community who responded to these surveys, and I think a concern that we see quite regularly on our helplines and through the conversations we have with patients and patient groups, is how genetic information will be used by insurance companies. So I popped up some quotes here, some of the views that our patients shared with us in the free text parts of our survey. And you can see that there is a real concern about how insurance companies may use this data. Interestingly, no participants in our surveys felt that insurance companies should access their genetic data. And further, we have seen the reasons why this is. Although there is a moratorium on the use of genetic data for insurance purposes at the moment, that could be subject to change in the future, and this was a concern as well amongst our patient organisations. So some final thoughts. From the perspective of the research that we've done, it is quite clear that patients value sharing their data. They know that genomic information has the potential to transform the healthcare system that we recognise today. Researchers are continually learning more about the genome and the genetic basis of disease, and more research will be possible. This will enable us to achieve a greater understanding of how our genes affect our health and develop new diagnostic tools, screening, methodologies, and therapeutic interventions for some conditions. And this can only have value for patients. What we know is that the relationship between patients and researchers is mutually beneficial. For many of the patients who have taken part in our studies, they have taken part in research already. So we have some patients who speak wonderfully about the 100,000 Genomes Project and the opportunities that has opened up for them. They do this because they have a hope of getting a diagnosis, or the hope of accessing a treatment, or perhaps undertaking a new care pathway that wasn't available to them before. And that is the reason why there is an overwhelming enthusiasm for sharing their data. At the same time, research studies do rely on being able to access and analyse patient data in order to make the advancements that patients so desperately want and need. To enable this relationship to flourish, patients who want to participate in research need to be able to share the genomic data and feel confident that it will be both stored and used properly. Trust is key, and for that trust to be maintained, patients must be adequately informed how and why their data is being shared and who it is being shared with. They must be allowed to provide dynamic consent and be able to withdraw at any time when they feel that it's necessary to do so. Data storage and sharing must be safe and secure, and it must continue to be for the purposes of improving the lives of patients. Thank you.
Thank you, Natalie. I might just make a small comment about the insurance companies. I think we're in for some surprises there. That there's a move afoot internationally for insurance companies to offer discounts for genomic information with a guarantee of no penalty for any genetic risk, because they understand that understanding the risk reduces the risk, which is in everybody's favour. So let's hope for a more positive outcome on that front. Our next speaker who's going to pick up the same theme is Phil Booth, who's the coordinator of medConfidential and a campaigner for citizens' control over their personal data, which I support very strongly. Phil.
Thanks very much. I don't have any slides, so I'll just be talking to the room. As you said, actually I think we concur very much with what Natalie was saying. I've been asked, however, to outline some of the risks and concerns that come up with particular reference to things that can go wrong. And unfortunately, as we've seen with a number of data programmes in England at least, things can and do go wrong with our health data. The most egregious example of this was the disastrously handled care.data programme, which some of you may have heard of, and also the associated revelations around how patients' hospital data had been misused for many years by the NHS body now called NHS Digital. This has led, at the latest figures, which I think were the latest statistics were published in September, to nearly 2 million people across England opting out of the use of their data, both hospital and GP, for purposes beyond their direct care. This is, of course, very relevant to research, which is a secondary use. Research by the Wellcome Trust and others suggest that these numbers could actually rise much higher. The latest projections we've seen, 25% or more, should there be another significant breach of trust. Yeah? So the stakes we are talking about here are actually very high. While you can deal with small percentage holes in data to maybe even 5%, once you get up around 20, 25, this starts to become difficult. So in the longer term, as we scale up, we have to get this right. Research, again, consistent with what Natalie was saying, shows that most people if the purpose is explained to them, and if they are given a choice, are happy for their medical information. And let's talk about medical information in the round here. There's, as I think you said, the genomic data, but also the linked health data, personal information. Most people are happy for that to be used for research, given those conditions. But if they feel their permission is not being sought, or if, as Natalie's slides show, they are not given proper information about what's being done with their data, then that dynamic shifts, and most are not happy. Yeah? Other conditions apply, of course, and these will differ across individuals and groups. And this is why medConfidential, since 2013, has sought to ensure a very simple thing, that every use of patients' data across and around the NHS and wider care system is consensual, safe and transparent. Without these things, they can't be. The very trust that we need, the public confidence and actually the individual medical confidentiality at the heart of the doctor-patient or clinician-patient relationship. So let's talk about some of the particular characteristics, and I realise this is probably more than usually aware audience. But let's talk about some of the characteristics particular to the data that we are talking about and why those may engage certain risks. I would like to dispose of the term big data to start off with. Yeah? It makes it sound like it's something new or different, and yes, indeed, the scale and the scope of data collected and processed is unprecedented. But this is data about people. It is therefore personal data, and as it's about their physical and mental condition, it is sensitive personal data, or as we now call it under the GDPR, special category data, and it is recognised as such and given special protections in law as such. And given the particular familial or kinship aspects of genomic or DNA and genomic data, that's likely to strongly engage things like Article 8, Human Rights Act, or ECHR, private family life. It's not just the individual, it's the family. So some of the old excuses, and I've heard the word come up, although in the context I need to be very clear here, some of the old excuses that have been used about removing or obscuring some of the most obvious identifiers simply won't wash. That has been an excuse, anonymisation, that has been used for many years. Yeah? Now, sensible measures must be taken, must continue to be taken, but we must be very clear. When you start to link this data together, certainly at individual level, where you have a sequence, as is most of the sort of data held by the NHS, a sequence of dated medical events, that is a fingerprint. Yeah? That is unique. That is highly identifying. And there is also the aspect of DNA that is inherently identifying, even though a data set may be called anonymous. The very reason why it's used in forensics is that it is highly identifying. And while you may doing, or researchers may be narrowly focused on doing research, an aggregation of such data is inherently highly identifying. Okay? So it's all identifiable data and must be treated as such. And I don't actually think I've heard any dispute around that at all. So what are those distinct properties that relate to people's privacy concerns? Well, this is not just your current state. This may be talking about your future state, your future health, the propensities to develop certain conditions which people may have attitudes towards. Your DNA actually changes very little over time as well, so it only increases the sorts of concerns that people have about the fact that it is highly identifying and traceable. Yeah? So there is also the fact that the value of the information that is contained within your DNA, and we don't know, this may be important or valuable in unexpected or unanticipated ways, that doesn't tend to decrease over time either. So genomic data is very different to a lot of health data, where like you have a test and that value declines over time. Your blood glucose level or your blood pressure. Yeah? DNA is only likely to get more and more valuable over time. And crucially, of course, your DNA, your genomic data, will contain information about your blood relatives. Yeah? your kinship, or, as some unfortunate folk have found out, not your kinship, with certain services that have been offered, probably ill-advisedly. Yeah? It's these things around, it's not just information about you, but about your family, which can make the issues around consent far more complex. And finally, but no less important, and one of the reasons why I guess we're having this meeting today, is evidence shows this is all really poorly understood by the public. Yeah? And things that people don't understand well, they tend to fear, or they tend to overreact to. Yeah? So it behooves us all to get people up to speed on understanding what this stuff is and how it may affect them. Yeah? Because when we're trying, as I'm sure we all are, in line with medical and with research ethics, trying to ensure that people make informed choices, that's what we're going to do. So quickly zipping through how these play out as risks. The most obvious, obviously, identification, re-identification, not just of an individual, but as has already been done by the police and law enforcement in this country and elsewhere, through inference attacks or what's called familial matching, the identification of people's family members. If you are giving something for, you think, beneficial reasons into a research database, but at some later date, the law enforcement agencies come to either the company or the body that is looking after that data and says, we want to check this out, evidence has shown companies will hand the data over. And that is going to have an impact on trust. Generally speaking, the creation of ever larger aggregations of biometric information, along with proposed and actual uses in crime detection, which I think is better understood, and border control, and with policies such as the hostile environment and a Home Office that is on the record, and a Prime Minister who, as Home Secretary, is on the record as saying any data set is fair game, we must bear in mind these responsibilities. I would say that means we must have strong statutory protections in place before we do anything, because we don't want a rerun of something like the NHS National Back Office, the Home Office Memorandum of Understanding, where Home Office were going hunting for people who they said had committed immigration offences through the information that the NHS held about them. That, again, would be devastating for trust. Misuse and abuse is not, of course, just linked to state surveillance and breaches, but it's really important that we understand, and again, public understand, that unlike your bank account or credit cards, a single breach is a lifetime event. You can't be reissued a new genome, new DNA. And of course, a big concern is depending, and I think as Natalie said, depending on who gets access, is the discrimination. I'm sure insurance is one, and certainly we've seen them have to legislate in the United States already, the GINA. That was because, and a colleague, Ed Hockings, Ethics and Genetics, did the research on this, women with the breast cancer BRCA gene were being discriminated against in work, because in America, obviously, you get your health insurance via your employer. So they're having to put protections in place for something where the genetic dice had gone. Anyway, being asked to wrap up, I would just, I guess, sort of say, return the basic principles that medConfidential seeks to embed. Consensual, safe and transparent. And let's try to keep trust together, have no surprises, as Dame Fiona Caldecott said, and not undermine what should be a noble joint endeavour. Thank you.
Thank you, Phil, very much. That's a good segue into our last speaker this evening, Dr. Sarah Chan, who's on the Ethics Advisory Board for Genomics England. Sarah is a Principal Investigator at the Centre for Biomedicine, Self and Society at the University of Edinburgh, and I'm particularly interested in the self component of that. Sarah, please.
Thank you, John. So tonight we're asking, whose genome is it anyway? So whose is it? Who does it belong to? And as Professor Mattick started out by claiming, we're concerned about our genome, so my genome, your genome. So I've taken the liberty of reinterpreting the question slightly in a way that's a bit more susceptible to analysis. So I'm going to ask, who does it belong to? Who owns it? Who owns your genome? Now, this question is not a simple one, because in fact we might mean a number of different things when we ask who owns the genome. Ownership might mean that we have the right to make money out of it, we have the right to profit, the right to buy and sell. Ownership might mean the right to exclude, the right to say, this is mine and you can't have access to it. It might mean the right to do things to it, so to use it as a tool or to manipulate it, to use it in certain ways. Or it might mean the right to read it, the right to have access to the information contained in the genome. So there are multiple different senses of ownership that we might be concerned about when we say, who does it belong to? Well, who can do any one or more of these things with it? Likewise, the genome can mean a number of different things. So there's the physical genome, the actual chemical, the actual chemical makeup contained within DNA, contained within your cells. So who owns that? Who owns the physical DNA? Who owns your cells, whether they are in your body or out of your body? There is the sequence, the letters, the bases, the A, C's, G's and T's of your genome, all millions of base pairs of it. There's the information that represents, so not simply the sequence, but the information that the sequence contains about you, about your identity, and about your health. And then, of course, we often talk about the human genome, so the genome of humanity, something that we share, and indeed we do share 99.8 or 99.9% of our DNA with everybody else. And it is that small, tiny 0.01% that's different between us, that makes us individual, makes us who we are, perhaps gives us states of disease or health. So we need to think also about the collective human genome. Now, I've asked who owns your genome, but in fact, as I think has become clear, your genome is not just your genome, because, of course, it reveals things about not just you, but about other people, about your family. But your genome can also reveal things about not your direct relatives, but about other people within the population. So, what we're really asking then, when we think about whose genome is it, who does it belong to, what we're really asking is the question of who can use our genomic data and how. What should we be able to do with it? As I think you've seen from some of the presentations tonight, genome research and genomic medicine has the potential to transform our understanding of human biology, our understanding of health, and in fact the way in which we understand and deliver health care. You may have heard the slogan that data saves lives, and I think we've seen some wonderful examples tonight of how genomic data can do exactly that. It can be used to save lives, to diagnose and to treat conditions that people suffer from. So the large-scale genomic data research that is being done here and across the UK is giving us new understandings of health and disease that can lead to prevention, to treatments and to cures. So in other words, your genomic information not only reveals things about you, about your family, but can also reveal vital information that can help to save other people's lives. On that basis, several bioethicists have argued that actually this means we have a moral obligation to participate in genome research. So essentially, the core of this argument is that if we can help others at little cost to ourselves, we probably should. So this is not rocket science, it's not even complex genomic science. It's a simple moral principle that if you can do something that could save a life somewhere, that could contribute as part of a collective effort to saving a life, to improving someone's life, we probably should do that. Now, of course, there's not absolutely no cost or no risk involved in participating in genomic and data research, and Phil's done a very good job of outlining what some of the concerns might be around letting our genomic data be used for research. Concerns around the security of the data, around privacy and confidentiality, and what sensitive facts about us our genomic information might reveal, about the possible misuses of this information, such as discrimination, and above all, perhaps concerns about who is benefiting from that information. So who is using your genome and what are they using it for? Now, I think it is extremely important that we take these concerns seriously and that we do our best to address them. I would say that although we often talk about data protection, what we're really concerned about is not specifically protecting data. What we're concerned about is protecting people from the possible misuses of that data and from the harmful consequences that might ensue. No data security system is ever going to be 100% secure. And I think that's just a given that we have to accept. There are tremendous security measures in place around the health and genomic data on which we're conducting research. So I work in the Usher Institute for Population Health Sciences and Informatics, and the sorts of physical and IT infrastructure that we have in place to allow people, well, physical, IT and ethics and governance infrastructure, I should say, to protect the data that people are working with. Researchers and research projects have to be vetted and have to be approved by scientific and ethical committees before they are even licensed to gain access to the data. The data is protected, although it is electronic data, it's protected by what they call airlock systems to make sure that data can't be taken in and out. And then it's physically protected as well. So to access this data, you have to go into a secured area of a secure building, and you have to go into a secured little box inside the area. You're not allowed to take anything in with you, so you can't take USB sticks, you can't take your mobile phone. And in that secure environment, you are allowed to work with the data, which has, bearing in mind Phil's cautions about the power of anonymisation, but you cannot simply just look at somebody's personal data and see who they are. So there are huge protections around data use, but at the same time, no system's 100% fail-safe. So rather than worry so much about data protection, we ought also to be saying, how are we going to protect people from what is going to be at some point inevitable? At some point there will be a leak. How do we then protect people? And I think in that respect, the sorts of developments that have been talked about in terms of protecting people from adverse effects of discrimination in insurance and employment and so forth are hugely encouraging. Okay, so there are not no concerns, but I think the benefits that are to be gained in terms of benefit to patients, in terms of potential improvements to health and lives saved, do impose on us some sort of, I suppose, some duty, some moral reason to support this form of research. Now, this is not to say that we should tomorrow pass a law saying everybody must donate their genome to research. What I'm arguing here is that it would be a good thing. It would be a good thing if all of us went out today and volunteered to take part in genome research. There's also the idea that's been talked about of the so-called social contract for biomedicine. In common parlance, this is often translated as saying rights require responsibilities. So we all benefit from the scientific knowledge that has been generated through research so far. We all benefit from public health care provided by the NHS. If we are going to claim that we have the right to benefit from scientific knowledge and the right to health care under our state system, we perhaps also have the responsibility to support that and to enable the NHS to provide better and more effective care and more efficient care by supporting the sort of research that's going to enable that. The slightly more technical way of saying that, I suppose, is that science is a beneficial social institution that we have a duty to support. So it makes society a better place. I'm going to go one step further, though, actually. It's not just that we have a moral duty to participate. Actually, we have an interest in participating. So Sarah asked the question of, would you be happy in principle for your genomic data to be used in medical research? I'm going to say, it's not just would you be happy to, we should be out there clamouring for our information to be used. So in fact, we should be much less concerned about how we stop people from using our genetic information, and much more concerned that our information is used and used in the right way. And I'll tell you why that is. If research is done with populations who are not us, people who are not like us, it will be less relevant to us. If all of the research that's done is done on populations of middle-aged white men, how relevant are those results going to be if you're not one of those people? So we benefit from research into conditions that affect us and people who are like us. We know that in clinical trials, for example, our knowledge about drugs in populations other than middle-aged white men was severely limited because nobody else was participating. So it's in our interests, actually. We have an interest in being included, and I would say, actually, we have a right to participate. This is a project that will require all of us. So you've heard talk about precision and personalised medicine, and the question about this is not just what's your personal information, it's where does that personal information place you in relation to others. Understanding your genome requires understanding other people's genomes too. So we're all in this together. Genomic medicine and research are inextricably linked, and we will all benefit from it, but only if we're all in it. And I think given that, the next challenge we have to face is how should publics be involved in deciding what is done with our genomes and making sure that what is done is what we want to be done. Thank you very much.
Thank you, Sarah. Now I'd like to invite all the members of the panel to come up and sit at the front so we can engage in audience questions and discussion.
Hi, my name is Alan Thornhill. I run a diagnostic service called Igenomix UK. I'm going to have my whole genome sequenced within the next 6 months, partly out of interest and partly because I want to put my money where my mouth is. What I'm interested to know from the panel is if I do that, how will that integrate into, for example, the current NHS system? Because my previous experience tells me if you go and do stuff privately, the NHS is not that interested, but I'm really interested to hear whether that's a good thing.
Right, we'll take that on notice and look for a couple of others to get the ball rolling. I've had my genome sequenced. I'm waiting for others. The one over here. Yep.
I'm David Porteous, involved in this kind of research, but I was really struck by Zosia's comment right at the beginning about the level of non-understanding about the basics of genetics in relation to personal histories and health trajectories and liabilities. So I suppose the question is, how are we best able to change that? How do we get more participatory involvement in the research process in order to secure what has been commented on by Sarah and Phil about having transparency and consensual approach to this?
Informed consent, understanding.
What does informed consent mean?
Right, well that's an excellent question, and we'll take one more and then we'll throw to the panel. Do we have another?
Hi, I just have a question about...
Can you identify yourself?
I'm Sarah, I'm based at University of Edinburgh and I'm interested in ethics and technology and issues around autonomy and agency and that. So my question was around, there was a slide on children and parents consenting to lifelong storage of electronic records. And my question was, how does that discussion come about? Can a child consent to that kind of information? How much control do they have once they come to an age of being able to make that decision? Can they withdraw information? I also, related to that, I noticed a mention of dynamic consent, and what does that mean in this context? Like, how would you actually implement that with genomic data? Yeah.
Very good questions. Okay, we've got three. By the way, people have thought about that quite extensively. It's reminding me we perhaps should think about a Q&A or sort of FAQ thing which accompanies genomic information, because many of the issues that are raised in this area and others actually have good answers, I think, to them. But I'll let the panel give you those answers. So we want to know how to get privately commissioned genomic data into the healthcare system, how to improve the public's understanding of what a genomic test is and what it means, and before they consent.
If I may, John, more than it's not about public understanding in the old-fashioned sense. It's about public participation in the shaping of research and stuff that I'm interested in. But I think it's more relevant.
Public participation, okay. And if we have genomic information, as I think we will, to replace the Guthrie test at birth for children, what information is revealed while they're a child, and what rights do they have when they reach maturity to control that information? Who wants to take one of those questions? Sarah's looking keen.
So I'd like to respond to David's question and Sarah's question, both of which I think raised some really excellent points. David, I'm so glad that you jumped in to say it's not public understanding, it's public participation, because, of course, the idea that, yes, it is true that many people don't have the kind of understanding of genomics that they perhaps need, but at the same time they are not empty vessels waiting to be filled with that knowledge and then be satisfied that we've done what we need to. We do need to think very carefully about how we engage publics in not just knowing about the research, but in having a say about what's done. I wanted to mention, I don't know how well known the Genomics England model is of having the patient panel, and that the patients who are part of the patient panel have input to governance and decision making at various levels. But I think this is a very interesting and potentially fruitful way of engaging participants in having a say about research. What we need to think about is how that model rolls out on a global scale. So we're learning things all the time from the way that we're engaging publics in participatory governance of genomic research here. When we start to merge data sets across different countries, what does public participation and participatory governance of science mean in these different contexts, and how do we then make decisions on a wider level about what's done? I don't have an answer to that, but I can say that part of the work that we're doing in our Centre for Biomedicine, Self and Society looks precisely at this question on how do we involve publics more in the shaping of research.
And what about the children?
So actually it's interesting, it was interesting that you linked that also to the idea of informed consent. We are never going to be fully informed to consent to the use of our genomes because we don't know what information is going to be revealed in future. How can I consent today to the kind of research that's going to be going on in, say, 20 years' time, when 20 years ago we hadn't even completed the sequencing of the human genome? I think in some senses this idea that we have to be fully informed about everything that's done is maybe not practical, but also not appropriate, because what research with participants in genomic studies and genetic studies has shown, for example here in the Generation Scotland project, is that what matters more to people, and what I think would matter most to me, is not knowing exhaustively every piece of research that was being carried out with my DNA, but knowing that the research was being done for public benefit, that the results were going to be used to benefit people like me, that individuals were going to be adequately protected should something go wrong. So actually that I suppose that I would have some sort of say, that I would have an appropriate decision about what sort of research could be done, and that needn't be by me saying yes or no to my individual data, but more the feeling that people like me are represented in decision making. So actually I wonder whether we should move away from individual informed consent as the most important thing. Don't get me wrong, it still is important to ask people what they want done with their personal DNA, but far more important is to say what's being done in general with our genome.
I'll pass over to the other participants, but I suspect it was also about return of information. And there's a general international consensus that information derived from a child's genome should only be returned where that information is essential to their management of their health as a child, or for something in the future, that all other information should be kept private until the child is of the age of consent. So only actionable information that affects the present or future health of the child that has to be actioned while they're a child would be released. And the Canadians have done a lot of work in this space in terms of the ethics around that. But would anybody else like to comment on either of those? Phil, and also Alan's question about...
Yeah, so really just to pick up on what Sarah's saying, I think the general tenor here. When I say consensual, safe and transparent, it's all a piece. Yeah, you can't have the one without the other. I think Sarah gave a very good description of what we called for, a safe setting, back in 2014 about the care.data scandal. But when it comes to things like consent and transparency, or why we say consensual, not just consent and transparency, we need to be feeding back to people what is going on. And one of the big things that all the systems that are being commissioned now, and many systems that have already been commissioned, have the ability to be able to say, who has used a person's data and for what purposes. This is just not being fed back to the individual, so they're being cut out of the loop of understanding how they have engaged in positive things like research. Yeah? So we've encouraged something called the Personal Data Usage Report. This may actually, as things roll out, this may actually join up with the genomic side of things, which would be that use of your hospital or GP data would be, you would be able to check, and the patient would be able to know who has used, literally, which program, which organisation has used their data, and over time, what have the outcomes been? Yeah, as you say, you can't know. It's too open-ended. We're at the early stages of an entirely sort of, I won't say entirely new, but we are at the early stages of whatever is going to be done with genomic data. So let's build the systems in such a way now that they give us the communication channels and the feedback loops in order to be able to either refresh or do dynamic consent, or certainly, I would say, most importantly, be able to tell people things that are true about what has been done with their data and give them meaningful choices. Prior informed consent is difficult. Dissent must be carried through, must be respected. You say you get to opt out, that opt out has to be meaningful. Yeah? Otherwise, again, you undermine trust. So it's sort of to try and think of it in the round, and we are going to have to innovate around the flows of information backwards and forwards between the system and the patient, I think, to build the sorts of ongoing trust that can make this into something that feels like a joint venture rather than something that has been done.
And Phil, there are a number of companies that are in this space that are going to using blockchain technology to guarantee the anonymity of the data, because you can't reassemble it without the permission and the key that's held by the individual, who can then give dynamic consent as the use of that data. It's quite an innovative model, which I think addresses most of your legitimate concerns.
And we're encouraging innovation in all aspects, because against these general principles, not, I think as Sarah said, there aren't going to be just techno fixes for this. Yeah, we're talking about healthcare is a techno-social system, and we have to get the whole thing working in the round.
What's intrinsic in this, I'd like to get to the third question, because there's a nice segue, is that for that model to prevail, then it has to be controlled by the patients rather than by the system. And so I suspect that we'll be looking both socially and structurally towards a movement that is actually direct to patient, but through trusted and trustworthy channels. Which brings us to the NHS. And if you have got the data, is it better to put it with an organisation that's got a health app that carries all of your data, and you insist the NHS gives it to you because you're legally entitled to do that? Is that a better ecology? Anybody got a feeling about, from the NHS point of view, how this might work?
Could I maybe just address the question about the NHS 100,000 genomes consenting process? So that is a consent where each individual taking part, it gives consent at the level of their ability to consent. So depending on somebody's age at participation and their intellectual ability, as in there is a definition of incapacity, for example. So those who have, it's about taking consent at the level of capacity that somebody has. So the conversation that takes place with a 1-year-old is not really the same kind of conversation as takes place with an 8-year-old, a 10-year-old, 14-year-old, and there are different consent forms depending on the age, but the premise of the project is that at age 16 every individual is reconsented to stay in the project or not, and the default would be they would come out if they didn't consent to stay in.
No one's mentioned it, so I'm just going to mention it. Our understanding of the model for the long-term storage, so you mentioned the Guthrie tests and what have you, so there would be a storage of a differential between a reference genome and your particular genome associated with your GP record. This is certainly what we've seen, presentations from going back as far as 2012, I think, on. So we're talking about some megabytes of data that would be associated with it, but we wouldn't be storing necessarily the whole genome within the medical record. The whole genome needs to stay somewhere.
Store the differences, that 0.1% of differences, because. But the reason I'm smiling a bit is because two things. One is that what goes on a medical record with consent will not be a VCF or variant file.
Yeah.
It'll be highly curated, well-validated conclusions from that this individual, for example, might have incipient familial hypercholesterolemia, and that the physician might look into that through other tests. So clinically useful advice coming out of it, but the basic data will be held somewhere else. The other thing is that we're starting to come to the view that it may be cheaper to store the DNA and resequence it than to store the information that comes out of it.
Yep.
So
this is the price slope is very counterintuitive.
well, we're not sure whether the storage costs are going to fall faster than the sequencing costs.
And the way we create the data and the way we can interpret the data will evolve and become smarter and sharper.
Yes, it will.
So probably in 5 years' time, you know, you if you get a floppy disk now, you probably haven't got any equipment to read it on. I think we'll be in the same space.
Yeah, it's going to be an avalanche of data, that's for sure.
So if you came to me as a patient, if you were referred by your GP having had a private genome test, my instinct as somebody running a genetics service is to think that actually it depends why you're asking. So if you have a highly genetic condition and you want to know whether that has a genetic basis and you have an interpretation of the genome you've gone out and got, and that interpretation is actionable, you have a mutation in BRCA1 that might be important for your daughter, or a mutation in BRCA2 that might mean you need prostate cancer screening. I would take you on. And I might check that result and check that the private company hadn't got the samples mixed up. But I would definitely check the interpretation of the variant, because in my experience that's where things tend to go wrong. And often, in my experience as a doctor, there is often over-interpretation of genetic variation. And I think private companies are getting smarter and better at it, but that would be my caution. And my third caution is what are, you know, it's all about why you're doing it and what you're going to find out. Okay. Does that help?
Yeah, I'm hearing scepticism, which is what I kind of expected. Yeah?
It's scepticism grounded in my, you know, the NHS's ability to deal with what you come with, okay? We can't see everyone who's well. So we need to focus on people with something actionable.
Can I follow up on that though? What if I don't have the whole genome test because I think I'm sick or I want to find out my risk of dying of heart disease or whatever, but because I want to be part of a population genome research project? Now, I was perhaps a bit disingenuous when I said to you all, hey, we should go out and participate, because at the moment, those everybody population-wide opportunities aren't quite there yet. We're getting there, but participation in the 100,000 Genomes Project was actually a rare and valuable commodity. You know, if you got in, if you got to be sequenced and you got to have your DNA studied, you were privileged in a way. So we are not quite there yet with we can get everybody's genome, but I think what we're moving towards with the incorporation of more and more genomic techniques into routine care is a position where we could all be eventually participants if we wanted to. But what option is there for me to get my genome privately sequenced and then say, here, put this into the Genome Project? Do we have any facility for that, and what would the concerns be?
There are certain options. Things like the Global Alliance for Genomic Health have these sort of beacon systems where if you put your data into an appropriately configured database, then I can, for example, find out that there's a person in your database who has a particular variant. I can't find out anything else about them, but I can at least see. So if your data is, I'm assuming, it's one person's genome is not, obviously quite important, but obviously 1,000 people's genomes are far more important. So if you're part of if your company, I assume, is sequencing more than just you.
Well, I'm not sequencing. I'm going to be a participant, but I'm saying we're all talking about this great sort of new dawn, but it's a new dawn for a very limited group of people. And I understand why, from a point of view of NHS resources. But if private companies, and forget about private companies, if I as an individual says I think there might be something useful in my genome for me and for the greater good, then why is there not a system that can incorporate it? And it seems to me there isn't a system that can incorporate it right now.
It depends who's doing the sequencing and who's got the data and whether they've decided to join in one of these collaborative enterprises. And one might say the enlightened sequencers are joining these enterprises, the unenlightened or the scared ones aren't. But there are obviously, as we said before, there's lots of good reasons why you don't want to join these things as well, because there's lots of misunderstandings and misconceptions about what it means. But I would say if you're going to have your genome sequenced, ask the people who are sequencing it whether they're prepared to share that data, whether they already share data in an aggregated fashion or in any other way.
I thought it was my data. I thought it was my data.
It did.
They need to buy into that too. Is that what you're saying?
I would say that a responsible sequencing organisation would do that, and I wouldn't want my genome sequenced by someone I think who didn't want to do that. Again, it comes back to this concept of for the greater good. You know, if I have my genome sequenced, it's for my benefit, but I think it also should be for other people's benefit. I would say, if company X wants to sequence it, I'll say fine, you can sequence it, but I want to know that you also got to share that information because I want other people to benefit from that as well. And share it in a responsible way, but I think that's fair enough.
Yeah, if this is your think about a donation. If you're making a donation, yeah, not every charity can, I don't know, process postal orders versus checks or what have you. Yeah, you give it through a channel that is already set up and structured to receive that type of data, and that is because of the nature of the development we're at. That's
a very good question. We'll set up a channel.
Which companies are working with which initiatives, isn't it?
For volunteers.
You know.
In fact, we actually see volunteers as an important part of the future.
You will want the sequence to be at the quality that you work with.
Yes.
And
yes.
have the additional
or it's in a bin that's
of
as
well.
Uncertain quality.
Yeah. So
yes.
that you can aggregate it and
yeah.
work with it.
One of the complications here is that if the data is taken into, say, Genomics England database, the NHS is, for very understandable reasons, unwilling or hesitant to act on that information unless it comes from an ISO accredited laboratory, that has certain quality standards. Yeah. So there are caveats, but I think in a research environment we should encourage volunteers. I know that what's his first name? George Church in Boston has started the Personal Genomes Project where people can volunteer. I don't think they need the permission of the people who did the sequencing. They just have to answer a questionnaire that says very clearly that they understand what they're doing, and then it becomes public. So it's almost a countercultural movement to the privacy movement, saying, look, some of us, not about me, but some of us don't care. We just put it out there.
But maybe it depends about what you want to do with it. So in the world I live in, people want interpreted genomes and effectively and correctly interpreted genomes. And I've spent 20 years talking to people about those variants of uncertain significance and trying to diffuse worry. So I suppose my caution comes from giving people genetic baggage that actually doesn't mean they have a risk, and or telling them about stuff we can't do something about.
I think we'll take a couple more questions just to broaden it out. The first one here.
My name's David Burt. I'm Director of Genomics at the University of Queensland. Kind of looking at the future a little bit. Karen, from what you were saying about today, because of cost and practicality and all the rest, obviously, geneticists focus on patients that come into the clinic with a condition, and you diagnose what's wrong with them, hopefully. But the future could be, for example, the gentleman in the front. You get your genome sequence, and I have no conditions yet. Or maybe I'm really young. I have no conditions that are apparent. You sequence that genome, you do an interpretation, and you predict that maybe when I'm 10 or something or other, I might have a condition that you can treat. And of course, if that would spread it to the entire population, a huge amount of sequence, well, let's just hypothesize that's going to be dirt cheap and all the rest of it. So we're in a different world. So how are we going to cope with that?
Thanks, David. Another question here.
It's a similar question, really, to David Burt. So I'm Tim Aitman. I'm with Zosia, one of the leaders of the Scottish Genome Project. And I do see there's a difference between sequencing the genome of somebody because they have a rare disease and sequencing the genome of someone for preventive medicine. And I wondered whether the panel sees there as being an ethical difference, and also whether the funding should be given to, where the balance should be on the funding of diagnosis of a rare disease, say, or preventive medicine.
Okay, so I have a third question here, and a fourth one there, and we'll cut off this group. Yes, those four.
Hi, I'm Filippo. I just started my PhD in human genetics in St Andrews. So actually I would have two questions. So the first one is, are GPs ready to use genetic information to talk with patients? Or is there too much information a GP can't cope with, or they don't know how to interpret it properly? And the second one is, there's a lot of companies that provide genetic sequencing and so on. Is there any collaboration, or do you think there must be any collaboration between NHS and those companies to pull together all the information as a shortcut, so to have access to much more data in a shorter time?
Okay, good. One more.
Hi there. My name's Niall. I have a bit of a global health background. I'm interested in what Sarah said about participation. I know that Genomics England has a lot of data sets within England, but have you collaborated internationally with any other organisations to share genomes so that you can start to investigate different variants in other populations to treat neglected diseases?
Okay, thanks. Well, okay, we've got two on the theme of how do we value, rate and segue to genome analysis for health management as opposed to diagnosis. And in fact, I think that is the critical question we have to address over the next 5 to 20 years. And then, can GPs cope with the information? And a second question was?
If the NHS is starting or is working with other private companies to put together all the data?
Not at this stage, is the quick answer as far as I'm aware. And then it was collaborating internationally. Well, perhaps I'll take that at the end. So, panel.
About GPs, I think the answer is fairly definitely no, they can't. I mean, most of them don't understand it. A lot of hospital consultants don't understand it either. I mean, it's not even the specialists. And I'm sure Zosia has probably got quite a lot of anecdotes about this. She probably can't. She probably won't tell you their names when she's tried to go to a fellow consultant and explain to them, as she says, things like variant of uncertain significance is a horrible thing, and we've all got tens of thousands of them, and it means what it says on the tin. But you try telling somebody that means what it says on the tin. It means we haven't got a clue what it means. You've got a variant. You've got 10,000 variants, and and they say, well, you had a test. Surely you've got a result. Yes, I've got a result. I've got a variant of uncertain significance. And they say, what does that mean? But I've told you what it means. It's uncertain significance. And you get a blank look.
Can I just interject there? No GP is going to say that to an individual because it's useless information. The GP is only going to say what has got a well-established genotype-phenotype correlation, surely. And so if the genome is interpreted well, the GP is going to get that information, and that's what will be transmitted to the patient, not a discussion about the esoteric nature of the other 3 million variants.
No, but even certainly at the moment, that information is going into genetic reports still. You might argue it shouldn't do, but it is still going into reports. Not obviously in the genomic context, but in the levels of these small panels and single gene tests.
Yes, so this really goes to the heart of the first two questions, is the emphasis at the moment on diagnosing sleuthing, the causative variant for a rare disabling condition, which are not rare at all, as you just put out. And by the way, I might add to that, about 1 in 8 of us will end up in hospital at some point in our lives because of a simple protein coding mutation. Things that will induce strokes later in life because of blood clots and so forth. So that's a big problem. But that's a different thing to looking at a genome, irrespective of why you collected the data, to look at what other information may be useful for the clinician to manage and improve the health of their patient, because that's what they care about. So, and that's the tension that I think David and Tim were addressing. Does anybody want to talk about that? moving from diagnosis to...
So if we think about prevention, I did my PhD looking at variants in the cystic fibrosis gene about a long time ago. And, you know, the issue there was that it was quite cheap. And lots of people thought, lots of pregnant women wanted to have the test, but it didn't get funded. And there has to be, in the way that the state funds testing, as opposed to people making their own choice to use their own money to have a test. That in the UK is based on tenets that were set up by the World Health Organization on screening. And that's about the benefit that you get for the investment that you put in. And the costs of creating a genome at the moment and then interpreting it and working, it's all, we've automated, and Genomics England has done an amazing job of scaling up automation of interpretation, but there is still a manual step that we heard about from Austin. And at the moment, the scale of doing that in the absence of something actually being wrong with somebody can make it very difficult to filter out what is a disease-causing thing from what actually is just noise in the genome. And so there are certain variants in certain genes where it's easy to say, yes, that's something nasty and we should do something about it. But there are many more variants in those same genes where we don't maybe know what they mean. And so you could falsely reassure someone by sequencing their genome and say, well, we didn't find anything. But they might still have a problem. And we don't even know the level of that uncertainty, I think, yet. So I think it's a work in progress.
For diagnosis, yes.
But for screening as well. Absolutely. Yep. If we screen BRCA1, if you've got an Ashkenazi Jewish variant, we can say with what level of certainty you have of getting breast or ovarian cancer. But if we find a missense variant in that gene or a stop codon in a funny place, in the absence of a phenotype, it's very difficult to predict what that means for the individual.
Let me give you a personal example, tease it out, because there might be a lot of variants of unknown significance, and I agree, it's difficult to know what they mean. But there are a lot of variants of known significance. And I found, to my surprise, when my genome was sequenced, that I'm a carrier for cystic fibrosis, the G551D version, which you'd be familiar with. I blame my mother. It's called the Irish mutation. I can't dig her up to check. But the surprise I got was that there was no family history. And I did the numbers and it was unlikely that it would pop up on a random basis. And my wife is largely of Caucasian ancestry, and she has a 1 in 20 chance of being a carrier. And we have 2 beautiful boys, but I don't know if we dodged a bullet or not. Because if she's a carrier...
So you didn't sequence her?
Well, we're not going to have any more children, so it's not really relevant at this point. Besides, it's a bit too expensive.
What about your children and your grandchildren?
Well, I've advised my sons, but by the time they're in a procreative state, I'm hoping that population-scale genome sequencing is available. And to be slightly more serious, because thalassemia has been almost wiped out in the Mediterranean by genetic testing. Tay-Sachs disease has been almost wiped out in all but the most conservative Jewish communities. And I think that prospective parents in future will know what genes might be damaged in common. So there's all these Damocles swords out there. Cystic fibrosis might be more common, but 1 in, 30 children roughly is born with a serious disability. I think that can be cut right back if parents know that they're a high risk and therefore take probably IVF approaches to avoiding the problem.
As also a carrier, my aunt didn't dodge the bullet of CF. The issue of there being prediction without certainty of treatment is one thing that we need to be very cautious about.
I agree.
Yeah? There is a big risk, I don't think anyone here believes that anything is a magic bullet. Yeah? But there's a big risk because of the low level of public understanding and the level of uncertainty, even amongst experts and specialists, that there is a general overclaim. Yeah? And so we need to be really clear about what it is that we can get at this. Yeah? As you say, the GP may be able to tell you or counsel you or refer you to a IVF counsellor or genetic counsellor if you turn out to have a particular variant. Yeah? But if we start doing whole genome stuff across a large number of the population, we're going to come up with a lot of information that is indeterminate and a lot of information that may be determinate, but about which we can do nothing.
Well, Phil, I agree, but I'd say it's pretty well understood that you would only return information where there's a high correlation of genotype to phenotype that's well validated on the evidence, number one, and number two, it is actionable, not just actionable, but practically actionable in terms of the cost structures.
Yeah, but we have a market already of companies that are getting people to send off their DNA and sending back a whole bunch of crazy stuff. There are even GP app providers offering 4 genetic tests as their business model for your accessing your GP data. You look at those tests and you go, what are you telling me about my diet?
All the more reason why we need nationally in the United Kingdom a trusted source that is there on behalf of the nation. And I'm hoping
let's not screw this up. Let's not screw this up. Yes. But there are signs that it could already be.
Natalie, would you like to chime in, please?
I'd just like to bring up something in relation to what you said. Expectation from patients as well, who are going into these programmes expecting a diagnosis and expecting a sudden change of, I now know what my child has as a condition, this will mean there'll be a treatment available, perhaps I'll get more support. That doesn't necessarily happen. In fact, we hear it quite a lot from families of, actually, we got the diagnosis, it was a string of letters and numbers. And nothing has really changed other than I can say it's a string of letters and numbers. So I think it's how you manage that expectation as well. It is very much a case of things may not change completely for your child. There may not be a treatment available. But actually, the value in that diagnosis for many families is just knowing. And I think that's a really important thing as well. But it comes down to how we communicate this with families who are undertaking quite a scary process for them, particularly when it's their child that they're talking about, and making sure that that information is as comprehensive as it can be.
Now,
I'm sorry, and we also have to be very clear that most people taking part in the project will not get an answer at the moment.
Half of them won't.
At the moment, only 30% are getting an answer.
There's a nicety here. The samples that have come into Genomics England and through Scotland as well are cases that have not been able to be solved by conventional means. But if you start from scratch and do a whole genome as the first-line diagnostic for a child with a serious undiagnosed disability, globally the diagnostic rate's 50%, so that adds the bottom 20%, which was relatively easy on the old technology with the hard bit, and it's going up. So I think there's a number of us, and I think the announcement from the Secretary of State reinforces this, that for any child with a serious physical and or intellectual disability, it's often both, whole genome sequencing will be the standard of care, so that at least 50% of those families will get an answer relatively quickly. And of course the hit rate will go up as we get more and more cases in the database. We'll get more and more statistical certainty that this variant is associated with this condition. Now, we've got 2 other things. One was about NHS cooperation with private companies. I think that, if I may, because closer to this, the short answer is no, but there is interest in government for its investment in genomics and related areas to help build a British enterprise in healthcare that may be exported to the world, and that may be done in partnership with private enterprise, but only on the condition that it adds to what the NHS can offer, does not detract, and allows British expertise to be exported to the world. So there are certain caveats, but that's very early days. So, but there is an interest, shall we say, in government to, particularly in a Brexit environment, for the UK to develop its wonderful expertise in this area, not just for the benefit of a better healthcare system here, but maybe to export that expertise appropriately to the world. So that's the more nuanced answer. The other question about global cooperation, well, some of, I don't know, Zosia, if you go to GA4GH, the Global Alliance for Genomes and Health, but it's a very active organisation that is working very hard to make public good out of the global efforts, whether it's in the United States, Canada, Australia, United Kingdom. It's mainly aggregated data. Once you aggregate the data, all you can do is do variant calls, just as was being explained about the Nomad database. So, but that's a level because there are strict privacy provisions, and each jurisdiction has different laws around this. So, I think everybody thinks we should be sharing data as much as we can for the common good, but doing it to protect, both obey the laws of the jurisdiction from which the data is generated, but also to protect individuals' privacy. Did you want to follow?
Yeah, I suppose there's a lot of genetic diversity, for instance, within the African subcontinent, and a lot of diseases which affect them exclusively. Thinking about variants within malaria, which impact people's, how they're affected by diseases. And I'm worried that genomics is going to go the way of pharmaceuticals and have a lot of emphasis on diseases which affect the Western world, but have exclude a lot of the problems that exist elsewhere.
Well, I think everyone's sensitive to that issue, frankly. Perhaps I should park over the ethicist rather than answer the question.
I think we are very aware of global justice in health and health research as an issue, and I think the point you raise, it's not just as simple as neglecting the diseases that affect people in low and middle income countries, but I think the bigger potential risk that we're facing is that those genomes are a rich resource to be exploited for research. The way in which low and middle income countries are brought into genomic research, I think, is going to be crucial. Are their populations seen as essentially a resource to be exploited by scientists who have the scientific, technological and healthcare capacity to do something with that? Or are they going to be used to capacitate the country itself to improve scientific knowledge, to improve care for those conditions which are precisely relevant? So, I mean, we're seeing in a number of countries at the moment development of national genomic research initiatives, and I think there's a real question about how will those countries be positioned with respect to the countries that are currently dominating the genomic research scene. Will they be brought on board as equal partners, assisted to develop capacity, etc., or will their data just be added in without respect to what's required to fulfil scientific justice? So I think it's a real concern, and we need to be aware of it.
I think implementation into healthcare is one issue. The other issue is having enough data to understand what genetic variants may underpin disease susceptibility in very diverse populations in Africa.
Yeah,
in this context, I mentioned, I think the Wellcome Trust is very active in funding these sorts of programs in the public good, and they have a global view. Perhaps it'll be a little bit delayed, but it will come, and I think people of goodwill are very keen to make sure that there is equity in this field.
And I think that's got to extend to scientific capacity as well as healthcare delivery. I mean, the sorts of disparities we're facing. So you've heard about the 100,000 genomes that we're doing here. I've been to Beijing Genomics Institute, where they have masses and masses, probably more now because I went there a couple of years ago. They've probably got 10 times as many of massive sequencing machines. And then I was in Ecuador, where until about 3 or 4 years ago, they did not even have a single DNA sequencer in the country. And I'm talking about, the old-fashioned ones where you do your PCR and then you feed your DNA in and you get this chromatograph printout. They didn't even have one of those in the country. So this is the kind of level of massive disparity that we're talking about, and actually how we bring countries up to the level required to participate fully in genomic science, I think, is a huge challenge.
The other thing I might add to that, Sarah, is that the Human Genome Organisation is very active in non-high wealth countries, and I think they're doing a good job there. But you're not going to get perfection, but we are trying to head in the right direction. Phil?
Just a quick reference, because it's very specifically something you mentioned. So this is Professor Sir John Bell's Life Sciences Industrial Strategy, which has been adopted by the government. This is not just a feeling inside government. This is an adopted strategy of government, looking at AI, genomics and other areas to try to stimulate economic activity and growth around.
Should have said interest in government.
Well,
yes.
Yeah. Yes. So this is a strategy. You can read it. It's published by the Department
Office for Life Science.
Office for Life Sciences. And I would encourage you to read it, because it is, actually saying this is looking at stuff that will be done with all of your data, all of our data. And this is, as you said, in the current environment politically, something where I think I read in the references given to us, we've actually just signed up, the UK just signed up to a cross-European coalition of people to share data, genomic data.
But it's anonymised.
Yeah, it's anonymised, but we have just signed up to something across Europe, which speaks to, well, who else is involved, but also what is going to happen when, if we are no longer participants in that endeavour. So, there are all sorts of issues that I would encourage people to keep on top of in terms of the politics and the economics of this, because it's not just an abstract thing. This is going to increasingly sort of affect us in our lives,
Could I answer? So Tim posed an ethical question that has not yet been answered, and I thought I'd give others a chance to speak before I jumped in. So, Tim, you asked about whether we ought to be prioritising sequencing those with rare disease with a diagnostic aim versus sequencing everyday people without an obvious disease for screening purposes. I think there's a clear sort of health justice answer to that question, which is that it is a more ethical use of our resources to focus first on those with the most need. So I think we are doing the right thing in focusing first on those with rare disease, with cancer, with other really seriously life-affecting conditions. But justice cuts both ways. So we need to attend to the requirements of those who are worst off. But at the same time, to the extent that being a participant in genetic research involves any burden, any risk, and we talked about those concerns, it would also be unfair if those burdens, those risks were borne disproportionately by those who are already unfortunate enough to have a genetic disease. So, in other words, yes, in some ways people are lucky to be able to participate, but we have to remember that there's a burden as well. Looking at, so this I guess relates to David's point about what does the world look like when everybody is sequenced. Actually, concerns about discrimination, privacy, etc., I guess start to look a bit different when we assume that the default is genetic information is there and is used. When genetic information on everyone is available, the question becomes much more, how do we treat people who are also genomic subjects? Rather than you and you and you, we have your genetic data, how can we use it? I'm going to keep mine to myself. So I guess what that means for what we do, if you're lucky enough not to have a genetic disease that you know about, you might not get sequenced just yet. But as the Genomic Medicine Service rolls out, I think the idea is that genetic screening, exome or whole genome sequencing will be more and more commonly applied in cases where it seems to be clinically indicated. And if you are offered that, and if your turn comes up and you're offered sequencing in a clinical context, then I think I would encourage everybody also to tick the box that says, and please use my DNA in research. So we should all eventually have a chance to be included, but the worst off first.
I might just add, the Secretary of State, as you know, a few days ago, made an announcement that the United Kingdom would sequence a minimum of 1 million whole genomes in the next 5 years, and has an ambition to sequence 5 million, which includes that done by the research agencies, including the Wellcome Trust. That, the motivation behind that, having been somewhat involved, is that would then obtain genomic data on about 10% of the population, going towards 100%, or at least of consented adults, assuming that they can see the value and trusting the curators of that data. So the real question is, if one accepts that genomic data is going to be extremely useful in healthcare, health management in the future, and I think that's widely accepted, the then question, which goes to your point, Sarah, is what do we do in the next 5 years? And so in laying out that, Tim, we've suggested that areas of unmet medical need should have first priority. And as a good example, which hasn't come up this evening, is people with chronic illness. Now, I'm not talking about with complex diseases, but who are always sick, what the insurance companies will call frequent flyers in the healthcare system. Now, that's from other jurisdictions because some people cost a lot more than others. We did a trial analysis of whole genome sequencing on people who are chronically ill, and found that, and these are on small numbers, 30% had monogenic mutations which would explain that chronic illness, including, and this explains it even further, mutations in immunological genes, which meant they were susceptible to chronic infection. Knowing that data means that you're in a better position to think about how to cover that deficiency and keep them out of the doctor's surgery or hospital. So we're on a journey from here to there. Rare disease is the first cab. I think cancer will be second. There are other sections of our population that will benefit early, but we are on a journey, I think, to having it as a standard part of healthcare. Tim.
And just responding to both of those points, the points that Sarah has just raised and the point that John has raised, getting the balance right between diagnostics for someone who has a rare disease and maybe applying genomic information to help prevent disease. Of course, the very first point of that is that if one is sequencing the genomes of someone who has a rare disease, and usually you're sequencing their parents as well, some of those individuals will have mutations or variants that might be actionable. And I would say that I'm very optimistic about this, because I think that information will be very valuable to those individuals and to the healthcare system. And the first thing to make sure is that we are able to cope with that information and feed it back where appropriate. And I don't think the resources exist for that at the moment. But as that information becomes more widely available, I think it's probably the first point that preventive medicine will really come into that genome medicine.
We're on the path there, Tim. You're quite right. With the families who've been sequenced, there is all sorts of other information. For example, we can identify those people at high risk for cardiovascular problems and intersect early to mitigate or reduce or eliminate the risk of those problems. The one we'll start with first, and we've already done this on 7,000 genomes, is adverse drug reactions. Now, people in the audience may not be aware that around 7.5% of all hospital admissions in this country and in other countries with similar health systems are due to toxic reactions to prescription medications. Think of coffee. Some of us can have a double shot and go straight to bed, others are wired for 24 hours. The same is true for your prescriptions, and if you happen to be a slow metabolizer, you can build up to toxic levels. It's also generally true that about 30 to 40% of prescriptions are useless to individuals because they metabolize them too quickly. So we have an enormous spend on pharmaceuticals which is not being of therapeutic benefit, and we have a huge problem in our hospitals of adverse drug reactions. I'm told it's not just 7.5% of admissions, but it's 95% of readmissions to hospital are due to toxic reactions to prescription medications. So we can predict those from genome sequences, and from my point of view, we have to show value to the people and to the health system. And then people, I think, provided we got the assurances, the privacy, the structures, people will say, okay, this is worth doing. And that's our obligation on us as a genomic organisation, is to show that value rather than just talk about it, which I think will make all the difference in the world.
PET is a charity, and I've got my special slide for Scotland, sort of stereotyped, and we do rely on people to support us. I usually, when we're in Manchester, it wears a Manchester United or a Manchester City kit. We really, we really do need your support. We only have 2 full-time members of staff and 3 part-time, so we'd like to grow and do more of these events and get some of the issues that we sort of talked about at the beginning around public participation, about trying to sort of make sure that people, if they want to, can engage in these issues and understand how they can engage. Because it's very difficult, and and some of the scientific terms, I'm not a scientist, and I get lost sometimes still after 10 years in the sector. But it's really important that that ability is there for people should they choose to take it. I'd like to do a massive thank you to Genomics England for providing the funding for this event and for working with us on it, and also to Wendy, I can't see her now, who's a great help in helping me identify great speakers to have up here as well. So that was really helpful. Thank you to Wendy. And we're back in Scotland after in the new year, just after Burns Night, on the 24th of January, talking about IVF, because the first Scottish baby is 40 next year. He's born to Scottish parents. It was a procedure done in England, in Oldham, by Steptoe and Edwards, but he's reaching his 40th, and we thought it was a great opportunity to celebrate with him. So you're very welcome to join us. That's a free event as well. And there are details about that and about our annual conference in your packs. So I'd like to thank everyone who's helped put tonight together, and I'd like to thank our speakers for sharing their expertise. And sort of, you know, it's been a very wide-ranging topic and discussion, this, and for, you know, really entering into the spirit of it. And there are, you know, we know some of the answers, we don't know all of them, but, you know, your honesty about where we are with our knowledge and how we're moving forward and the precautions that we should take as we go forward, I think are really valuable. And some of the things that were said sort of reminded me of some of just the basic principles of running a charity, and that's about good stewardship and good communications, and those that, you know, are very central to this as well as the science. So thank you all. Thank you, speakers, and thank you, the audience, for coming out tonight and for your really great and thought-provoking questions and comments to the panel. So thanks, everyone.